Seems like we have a little problem though with the display. Okay, great. Let's get started. Welcome everyone to Machine Learning for Bioinformatics. So from this lecture going on, we're going to enter our first module about functional genomics and mostly about regulatory genomics. And we're going to learn both about part of the biotechnologies and the important assays. And the data and understanding how we get the type of data that it's needed for this kind of research, and we're also going to learn a little bit of the biology behind it. Because our class has a kind of a mixture of students with pure CS background and some bioinformatics background or biophysics or biochemistry background. So please bear with me if some of the material sounds familiar to you. You already take like advanced biology, and that's totally fine. And if I'm teaching anything wrong, please raise your hand and just correct me, because I'm also learning a lot of this during my PhD as well. Cool. So today, so I think for each of these modules, we're gonna arrange it in a way that we kind of start off with some biological background introduction, so everyone gets familiar with the like the nomenclatures or terminologies and the key, like kind of the keywords and data structures that we'll deal with in the first lecture of the module, and then we go into some technical details of the machine learning models, and then we talk about specific extension and advanced applications to the topics related to that module. So our first module is about regulatory genome, and as I showed previously in the central dogma, right, a lot of the functionality of our body, a human body, is actually coming from this process where all of the information that's needed to empower life is stored in, is coded or stored in our genome, and from the genome we have this translation process, transcription and translation process to make the proteins that's functioning. But if we think about our cells, right? There are probably trillions of cells in our body, and like tens of thousands of cell types and different functions they're performing, and they're really complex system, and they need to also react to the ever-changing environment. Like if you get disease exposures, your immune cell will become different, and they'll be active, and they will function through different, very complex cellular processes. However, each of us only have almost like one genome. So all of this information that's controlling all of this complex process is stored into the same kind of genome, which is basically the mystery that's powering this entire kind of field, and is what our research is about, like basically understanding how our genome contributes to or dominating these cellular functions and different cell types, differentiations, and how we can intervene or try to make changes to correct abnormalities in our genome. So as the basics, a lot of the genomics are related to the study of either RNA or DNA, which both are kind of a nucleotide, polymer of nucleotides. So each of them has 4 types of bases, or we can call this nucleotide, or sometimes abbreviated as NT. For DNA, it is a kind of it has this double helix strand structure where 2 of the nucleotide will have this base pairing effect, such that they can get this stable double helix structures. So that means that we have 4 out of the 4 possible nucleotides, they are complementary between them, such that T will always form the bond with A, and C always form the bonds with G. So we call each of these pairs of nucleotide base pair, or BP. So sometimes you will see these abbreviations as the kind of the unit to count how long or how many base pairs there are in a sequence. Similarly, we have this RNA, which are ribonucleic acid, which also have 4 basic unit. So I think the 3 of them are shared with DNA, and the only different is the U. So and we kind of know that during the transcription and translation, during the transcription process, our DNA will kind of open up, and then the RNA will start to form, which kind of transcribe the information from our DNA into the messenger mRNA, and then the mRNA got translated into the protein, right? So if we look at this, how we represent our DNA and RNA, actually we only need the information from one of the strand. So basically one unique strain of T, C, G, and A, these 4 words, these 4 letters, can uniquely represent the information of a gene. The other thing about this nucleotide is actually, you can see that this bond forming between consecutive nucleotide, through this reaction, each of these nucleotide has two ends. We call it 5' and 3'. So these 2 ends are asymmetric. So you can see that for the 2 strands in the double helix, one of them is starting from 3' to 5', where the opposite side is starting from 5' to 3'. And the transcription always happens in a single direction, usually from 5' to 3'. But we have kind of 2 copies of the same information, but in a complementary composition. And both of them are actually encoding very similar and useful informations. So one way of representing sequence is using one-hot encoding. So we can imagine, you can kind of think about this sparse matrix of 4 by L. Well, L is the length of the sequence. And in each position, only one of them will be assigned a value of 1, and the rest of them will be assigned a value of 0. It is kind of commonly used in representing sparse or discrete features, like even sometimes in text or other discrete features. And the second thing about our genome is it's actually very big. So think about the huge amount of information that's encoded in our genome. And think about how small our cells are, right? So actually DNAs are not always in the most flexible and open position, because if we kind of extend all the DNA, if we just stretch them such that like all of our DNAs are open, it actually has a length approximate to 2 meters. But our cells are like much more smaller, and our nucleus are also like much more smaller in the kind of nanometer ranges. So this phenomenon is kind of called DNA packaging, where basically our genome information will never always exist in this kind of free and open format. It will be compressed by several stages. So firstly, some of these DNA helix will kind of be will be organized and compressed by centering around these histones, which are proteins that in our nucleus that helps organizing and form these small units called nucleosomes. And nucleosomes then will also will have a lot of this nucleosome, and they will also pack together to get the kind of fiber-like structures in the chromatins. And then this is still like just a small fraction of our entire chromosome, and you can see our chromosome also has this linear structure, which also has a kind of like open state and a more compressed state. So they're further condensed into forming the chromosome, and the chromosome actually also have different phases, which sometimes they exist in a more compact form, where you can visually like see these kind of X and Y-like structures, and sometimes they're like just kind of evenly distributed in the space. So we can see that actually this compression rate is super high, and our chromosome is 50,000 times shorter than if like extended DNA. So a lot of the compression happening here is also a subject of study in the current kind of advanced regulatory genomic science. So one thing we would like to highlight is that since things are in this compressed form, it cannot be, basically the transcription cannot happen if things are always in this compressed form. So so in order to enable or start the transcription and translation process, part of the genome needs to open up. So there's this concept called accessibility or openness of chromatin, which is actually the first step of kind of the necessity of DNA being transcribed. So the chromatin accessibility is one, so it's kind of like one important steps in this central dogma. And secondly, if we take a look into actually the gene, I think this might be known to some of you that not all of our DNAs are genes, or not all of them are functioning as a gene, where we kind of think about gene as the part of the DNAs that will actively engage in this transcription and translation process that actually kind of doing this protein, generating proteins. However, actually, when we actually take a look at our DNAs, not all of them are genes, and actually also a lot of them are we don't even know what they're doing. So, for example, we call this the part of the genome or DNA that actually code the protein information coding genes, and the rest of the DNA is non-coding DNA regions. And it turns out that actually from 98 to almost 99% of the DNAs are belongs to this non-coding DNAs, where like a huge chunk of them we actually don't know what they mean at all. They could be like totally nonsense, or there can be some hidden grammar in this part of the DNA. So this kind of it's like a hidden gem in the realm of biology or bioinformatics, where a lot of this advanced study is really trying to figure out what this 98% of the DNA is doing, besides just being translated into proteins. All right. So how many of you know how many genes do humans have? Or you have an estimation of how many genes humans have? Any guess? 30,000? That's actually very close. Yeah, I think about from between 20,000 to 30,000, and we're kind of still figuring out the exact number. But how many genes do you think fruits have, like grapes have? Raise your hand if you think grapes has more genes than human. Raise your hand if you think human has more genes than grapes. Yeah, we're screwed. We actually have much fewer genes than grape, right? So, and we actually, there are a lot of, I think basically, this is kind of the interesting feature about genes, and usually, like, you can see that the length of the gene not necessarily contribute to more complex function, and the number of genes also not necessarily contribute to, we cannot say like we're a kind of advanced creature than grapes, but obviously we're conducting like more complex functions than grapes in some activities in areas, right? And so it's really, what's really important is really not just the number of genes, but how we can actively make use of these genes and use them to kind of control the cycle of lives in a very efficient fashion. And if we could take a closer look of the genes, where if we consider the parts that kind of we already know that is related to translation and transcriptions, a lot of the common structures in this kind of gene region comprises this open reading frame region in the middle, which is basically what the parts that get transcribed into mRNAs. And then in the upstream or downstreams of the open reading frames, there are very important regulatory components. For example, there are promoter genes. Well, promoter genes where basically we believe triggers the start of the transcription effect, and it's where it signals the important proteins to bind to the DNA and starts transcriptions. And not just that promoter is important. In the further upstreams, there might be in the very distal region, which may not connect, located very close to our genes. There are also these enhancer regions which try to enhance or accelerate or basically triggering the actual start of the transcription. And on the contrary, there are also silencers, which basically act, repressors or silencers, which act totally in the opposite direction of enhancers. Of course, these both silencer and enhancer, they can locate in both upstream or downstream of the gene, so there's still a lot of the complexity here. And the silencers will try to repress the expression of the gene. And there's also terminator, which signals whether our translation should stop. And a lot of these components, it's easy to visualize it in this way, but if you think about how they look like in the genome, they can be anywhere, upstream or downstreams, and they can be also located in the different relative positions if you also consider the 3D structure of the DNAs. And in this open reading frame, although a lot of them are being transcribed into mRNA, still not all of them code a protein. And this is another important process in transcription called RNA splicing, where basically our ORF region contains some DNA codes that called intron, which are basically sometimes not, even though they're transcribed into pre-mRNA, they will eventually not going to be translated into part of the protein. So that's why we call it intron. And then the exon are the part of the DNA are actually translated and become protein at the end. So there's still one step of complexity here, where kind of that non-deterministically part of them will be spliced and cut out from the messenger mRNA before passing into the next step of actual translation. And this is also a very kind of mysterious steps where some of we understand part of it, and we're also having tons of unknowns of how this splicing is happening. And sometimes we also know that in different cell types, for the same type of gene, you could actually translate into different types of polypeptide and proteins just by doing a different alternative splicing. And there are also some hidden codes in our DNA that try to control this behavior, which also triggers some area of study. All right, so now we kind of have a brief high-level understanding of how DNA is engaged in this regulatory process. Any questions so far? Cool.
Is a gene only called a gene if and only if it's in the coding DNA?
No, actually genes has more, you can see that only the exon we call them like coding DNA, and everything else even in this framework are not coding DNA, they're non-coding, but we can still consider them as gene, as long as they can actively participate into transcription process.
So I know that there's like one RNA can match many different genes. So how is that calculated? And is it possible that if we have a lot of RNAs in, many of them is going to lead into the same ones, or there's like a many-to-many relationship between the RNAs?
Yeah. So you mean, for example, if we have two, I think usually the information usually if we transcribe into a different type of protein, then basically the coding information should be very different. I guess what we're talking about is when we have like multiple copies of similar gene, which actually happens a lot in our body. We have different copy numbers of the same gene, or the different variants of the same gene, like one from your dad and one from your mom. So yeah, I think that's definitely a very interesting question, and it will also influence how later in the course we'll talk about mapping the sequencing reads back into the reference genome. But even though a lot of the regions are similar, but there's still, if there's still like tiny variants in some of the DNA, and we will be able to capture those small variants by cutting things down into smaller reads and smaller fragments, such that fragments is sensitive to those difference in like small variants in the gene. And yeah, I think there might be those ambiguity, as you said, that it needs to resolve, and a lot of the modern bioinformatics tools kind of are already doing those complex computation to resolve that. Yeah, that's a great question.
I see there's like a gap between the promoters and like where the first exon begins.
Mm-hmm.
How
You mean this part and the Tata box?
The promoter, the gap in between, I think the first exon. How does the transcriptase know when the promoter ends and when the gene begins?
Right. So I think the part that's engaged the RNA polymers are quite, the pattern is quite fixed, like the Tata box, right? So that's usually signaling that the actual starting of the coding, like the ORF region, is very close. But I think the gap between that Tata box and other control elements are actually quite flexible, and there are a lot of spatial patterns we also need to learn. That's why we're using these machine learning techniques to see like all different possible patterns under the regulatory process of different types of genes and cell types. Cool. So now that we have kind of some understanding of that, that the DNA itself actually helps regulating the transcription and translation of itself, but there's more that's actually engaged in this process. So besides just DNA, there are also proteins and actually other small molecules and sugars that provides additional information, which also kind of provide another set of language or vocabulary on top of the DNAs, which they're basically mostly like known types of, we'll call this epigenomic states or epigenomic marks. And there are mainly three types of epigenomic mechanism of generating epigenomic marks that were known so far. The first is, of course, DNA accessibility, where basically the part of the genome will kind of open up with engaging in different kind of this DNA, these enzymes that participating in trying to open up the DNAs, which we'll talk about called DNA accessibility or chromatin accessibility. The other thing is called DNA methylation, which is mostly known to happen on these C and G nucleotides. We'll call them CpG. So if you have C and G, consecutive C and G on one side, it will have the G and C on the opposite side. And it's known that sometimes this C will have additional methylation, and basically a methylized C will be differentiating from a C without methylation. So sometimes that will impact whether or not a protein will bind to this DNA and start regulating with that. And the third type of epigenomic marks is called histone marks. So histones are these proteins, or actually a set of proteins that form this bead, which kind of organize where the DNA starts kind of to get around with these beads to form these nucleosomes. And actually, there are a lot of one histone consists of three sets of histone proteins, basically H1, H2, and H3. And for each of these, H1, H2, and H3, we get 2 copies of them. So you actually have 6 proteins that are packed together as histones, and they will try to organize the DNAs. And you can see that each protein in these histones also gets some slightly different modifications, which is done through the post-translational modifications, where you can add kind of different functional groups and sugars or methylations to each residue of these proteins. So usually how we read this is H3 means it's a type 3 histone protein, and K4 means like at the 4th residue of this protein, there is some modifications, and also at the 3rd position of it has some methylation happening. So you can see a lot of combination of these modifications can happen on our histones, and therefore it creates like a huge number of additional signal that we need to track, that actually known to be also impacting how our genomes are being accessed, and whether or not something will be able to open up or not, and whether something will be able to start regulating transcription or not. Any questions so far? Cool. So this is just a kind of a example of like what actual like genomics data could look like, and we'll go into more details on how these data are generated. But you can imagine like we have these x-axis, which is the kind of the position, each position in our DNA or genome, and then on the y-axis we'll be observing different level of activity, where these activity you can consider them as like intensity or frequency of observing certain activity. or certain events associated with that position. And well, we're actually having very advanced tools to probe into each one of these very delicate type of histone marks and DNA accessibility signals. So this kind of interesting work earlier in the, it's not like that old, but it's more considering the current deep learning based model, this might be a little bit old, but we can basically think about these features, these marks and their intensity at each position as the features of that position. And we can try to categorize our genomes into, or annotate our genome into different states by kind of learning a model that maps from these features to hidden states, which we can think about all different kinds of latent models. But earlier in the days, there's what we call hidden Markov model, where we try to assume that our chromatin has this kind of Markov chain of different states, and we try to learn how many states there are in our genome, and how the genome function translates from one state to the other state. And they actually found very interesting patterns, even with this kind of simple tool of like hidden Markov models. So now that I think one student asked about the concrete mechanism of these kind of transcription factors, it's actually quite delicate, and a lot of interesting interactions happening here. And what I'm talking about here is the part that we know, but still like there could be a lot of unknowns. So basically on the upstream or sometimes downstream of our gene, so at the exact beginning of our gene, there's a promoter region which have this Tata box here that's kind of signaling the binding of RNA polymerase, but it also has part of the other region that can be a very different transcription factors, which kind of interact with the enhancers in the further distal upstream of gene. And these enhancers or activators, they actually engaged some transcription factors. Oh, by the way, I have not introduced transcription factors, but these are all kinds of proteins that binds to our DNAs and participate in this gene regulation process. So every protein that engaged in this binding and regulating process, we call them transcription factors, or TFs. And they're known, there's tens of thousands of known transcription factors that we've studied. And how it works is it's kind of, the enhancer will bind to the enhancer region of the DNA, and also the promoter will engage the transcription factor related to promoter. So there might be some DNA bending proteins or some other proteins that helps the DNA to kind of wrap around, and such that the distal region of enhancer now get contact with the promoter, and they will basically further activate the promoter and help accelerate this process, or triggering this process of RNA polymerase binding and signaling the RNA polymerase to start doing the transcription. And so I won't read all of these, but just for reference, there are different types of transcription factors. Besides just promoter, enhancer, and repressors, there are also other types of TFs that are necessary for the transcription. Some of them we don't even know how maybe they modulate the transcription. But one key component of this regulating process is the fact that the DNA and the protein is interacting with each other, and the biophysics between the protein and DNA is triggering these different types of response. So we can basically say that transcription factors using these domains, we call DNA binding domains, to recognize a specific DNA sequence, such that it only binds to the DNA when it sees certain sequence there. And then it will have this very specific way of regulating which part of a gene it wants to interact with because of this DNA or sequence specificity. And there are a lot of different biophysics that's happening here, but just remember that because of this complex biophysics between these proteins, the amino acid side chains of these proteins and the backbone of our DNAs, it requires highly, it requires a lot of complementary, for example, shape complementary and base complementary to actually make the binding happen. This also partially contributes to why the binding are considered to be specific. And this is also why we believe that we can learn the signal from the sequence alone about whether or not some protein will bind to it or not. Any questions so far?
In the previous slide, I think there was like the example showing that if the A was changed to C and
Yes.
having
Yeah.
explain about it.
Oh yeah. So basically, if we have like so you can consider these two populations being one of being healthy, the other being kind of population with disease. And what we can do is we can sequence their genome, and we'll find that like one of them actually have these mutations in this DNA binding region, TF binding region, which result in the means of binding with this TF1, which could be very detrimental, because without this, maybe this TF1 is an enhancer, and this person will not be able to express the gene because of the lack of this particular transcription factor binding. And the actual mutation might happen in multiple positions, which can combinatorially cause a more complex effect. And yeah, so basically what we're studying is we try to find these examples of healthy population and population with disease or different types of phenotypes, and understand the difference of their genome, and combine that with other types of tools that I'm going to introduce today to understand the mechanism behind the disease. Cool. All right, and also like I did mention is that transcription factors or DNA regulating modulators may not be just proteins. There are also some studies that also like kind of show that, for example, like the epigenomics also matters, and also like the microRNA sometimes also have some functionalities in participating in this regulation. So the molecule space of the molecules that are engaging in the transcription factor or engaging in the transcription is more complex, and a lot of interesting open questions needs to be defined. Okay, so now that we believe that there's certain information in the sequence alone that can decide the binding, we actually the study of gene regulations actually a lot of them start from these discovery of things we call motifs, which are these short DNA sequences which reflect certain pattern that decides the DNA binding specificity. So I will talk about some example of motifs later, but for now we just kind of have a high-level idea that like some short consecutive sequences of the DNA may have certain patterns that is predictive of the different transcription factor binding. So now that we think about the entire genome, in the higher level regulatory process, it's actually happening at every different levels. There could be enhancers and promoters, but also like in the higher level in the post-translation process, there's also this splicing and other types of signals that are signaling changes to the protein. So if we think about our gene being kind of a complex Turing machine, you can think of these genes being or a Turing machine or kind of a machine. Then our genes which are kind of the coding regions that are transcribed into protein can be considered as like parts, and all of these regulatory regions can be considered like as a circuit. And there's also a combination of signals that are changing like all the time that kind of activate this machine to function differently. And we can think of this higher level language of DNA or grammar of DNA that start this sequence of events which is programmed in some way that's related to all of these parts in our genome. Yeah, this is yet another example of how the changes in the DNA sequence and motifs can result in different types of phenotypes. In this case, it's the FTO obesity. Cool. So now that we know that the gene has a lot of different functions and marks, how are we going to query about these? How are we going to probe about these activities, right? So there are actually a lot of cool technologies in the past, I would say 10 years or 20 years, that actually enable us to become where we are today, where we have so many data that opens up this new area of like a modern bioinformatics, especially empower the study of deep learning because of the amount of data is just enormous. There are several of them are very important techniques that I'm going to introduce briefly today, from just how we sequence our genes to how we use this sequencing technology in combination with other microarrays and high throughput assays, such that we can probe these different functionalities. And if we have time, we will also talk a little bit about gene expression and how we kind of capture the signal of how much gene are expressed and what kind of dynamics they have. Okay, so next generation sequencing. How many of you already know about how the sequencing is done? Cool. So I will still talk a little bit about it then. So basically, well, in a high level, if we have like a very long, if we want to know the sequence of our genome, our genome has kind of millions of base pairs, right? It's very difficult to go through them like one by one. So the high level logic here is we try to break down our genome or the things that we're interested about into these small fragments, and then we can prepare this small library of fragments. And we try to grow these fragments because we need more statistical power when we do sequencing, because there could be errors. So if the more copy you have, you kind of know the uncertainty and accuracy of your sequencing results. So we go through this process of growing these DNAs through what we call PCR. And when we get abundant copies of these fragments, we pass them to this plate, which basically is having us, well, there are different details about how the sequencing is done, but in general, like you can consider that the plate has this sensor, basically a fluorescent proteins that will recognize a particular base pair type. And once they recognize that base pair type, their fluorescence will tell us what type of DNA nucleotide it is. So starting from this library construction, as I mentioned, we will break down our genomes into these small fragments, and we will amplify them through this polymerase chain reactions, and then we will add some adapters to the into the two side of these fragments, because these adapters will kind of first just fix this DNAs on top of the flow cells. And the other thing is we'll signal particular types of enzyme or proteins to recognize that there is a sequence here and you need to start sequencing it. All right, so once we have all of our fragments in our library, the actual sequencing happens on these things called the kind of flow cells, where each DNA, small fragments of DNA is attached to the flow cells. And actually, we can attach multiple of them into one of these cells, and we have this sequencing, well, I would say like reagents that basically doing base by base reaction or recognition, where as I said, there are four types of reagent where one of them only binds to recognize one type of nucleoside. Once it recognizes it, it will the fluorescence of that reagent will shows us what type of nucleotide it is, and so at each step we take a picture. I can consider that as like taking a picture of the flow cell, such that you can see for different locations what the nucleotide type is. And when we wash away this fluorescence, and then we go to the next base pair, and we do it again and again. So by repeating this for the number of times that equals to the length of the DNA, we will get like L number of pictures or scans of our cells, and then we'll be able to tell like at each position of the cell what sequence it is by piecing them together. It's kind of a high-level demonstration, but I'm sure like some of you might know far better than I. But I think this is enough for you to kind of understand the mechanism, right? So, any questions so far? Cool. So now we have this reads. Now we have this kind of sequence of short fragments of that we have in the library. We call the sequence that we read through this process reads. So if you see reads, it means kind of this short sequencing result of each of these small fragments. And what we're doing next is to map these reads back to the genome, such that we know how we kind of want to piece them together to recover the original sequence information. So although like each of us looks very different, actually our genomes are not that different. So so basically we can think of, if we know one person's genome, a lot of the genome is kind of the same with the other people. So you can use this person's genome information as a reference, and just to try to map the relative position of these reads, right? So that's kind of mindset there is we're mapping these reads back with some reference genome, and there are certain bioinformatics algorithm that try to resolve this ambiguity in mapping different reads. And because we're breaking the sequence into different parts where it could be overlapping with each other, you get this kind of duplication of, like the small overlap will tell us maybe these two reads are consecutive to each other. And by having a lot of reads, we'll be able to resolve all of these ambiguities. So there's a concept called like depth of sequencing. Is basically like how many reads you get from this sequencing process. So the more reads you get, or the deeper you sequence, I think the more information you get, and the lower noise, and a lot of the noise will cancel out, and you get a more accurate readings. But I think in some certain cases, we will do this de novo assembly, where we only rely on these overlapping regions to assemble the sequence without any reference genome. Sometimes it can happen like within a single gene, if we know there are enough overlap. But I think the high-level idea is at the end you will get this these reads being mapped to each position, different positions of the genome. So in the sequencing round, the outcome is to get the sequence. So we'll be getting this recovered whole sequence. But in some other assays where we don't care about getting sequence, but rather care about position of certain activity, you can treat of these reads being kind of a mark of whether something happens in this position or not. And there are and when we do the sequencing, we can, because we kind of, you only have one ends that attach to this foci, so we have basically to pick an order of how you decode or how you sequence. And as I mentioned, there's a 3' and 5' for ends of a single DNA. So there are single end sequencing where we always start from maybe the 5', and there's also pair end sequencing where you can read from both ends together, and they may or may not overlap, but this gives you kind of the information in the reverse direction, which you can use to kind of correct the mistakes that happens in between. And there's also a way of speeding up your sequencing speed and getting longer sequence read, because now you can read from both sides. You basically double the effective size that you are sequencing. And a lot of time we don't want to just do one sequencing on one type of cells or single samples. We actually want to make use of this high throughput capacity. And a lot of people do is called multiplexing, where you mix samples from maybe different cell type or different human or different libraries. But as long as you can tell the difference from them by adding some special marks, which we call barcode, basically attaching this barcode into your gene, into your library, you can always separate the two libraries out later by just reading these barcode. And there's also a complex and bioinformatics tools involved in designing this barcode, so it don't overlap too much with the information that's important to your sequence. And basically, you try to find the sequence that appears the least in the things that you care about, so you don't mess up with the information you want to read. All right, so now that we know how to sequence a DNA, we can actually think a very different way of using this tool creatively by combining it to other types of assays, which tells us more about the function and other than just the sequence, right? So one important such assay is called ChIP-seq, which is what we use to study all of these interactions between DNAs and proteins. DNAs and proteins that can involve both transcription factors and also like histones. And we can use that to also study histone marks, for example. But so in a high level, I think this is a cuter image. Basically, what it does is when we have a fully functioning genome where we already know that some of the protein is binding to some of the genome, and we kind of add this glue, biological glue that cross-link the protein and genome together. So kind of the protein that's already bind to DNA will be fixed at the position that it binds. And then we cut the DNA into small pieces such that such that in the pieces where you will get the sequence as well as the protein that binds to the sequence. And then we will use different antibodies where antibodies will only bind to some types of certain types of proteins. So we can use antibodies to pull down the sequence that attached or bind to a particular type of DNA. And then we can do the sequencing. We can use the sequencing technique to sequence all of these short fragments we get from the particular that are attached to a particular antibody, and we can map them back to our genome, right? To basically get like how many reads are coming from this particular region of your DNA, which kind of also in a high level tells us information about whether or not how frequently you can see a protein binding to this region of the DNA. So yeah, this is the kind of high-level idea of ChIP-seq, and it's actually very, very effective and provide a lot of interesting data in the early days of modern like genomic studies. And you can also do kind of different. When you sequence, you can choose different directions, and you have a positive strand and negative strand. And you'll be able to study strand-specific binding because although like we can know that the two strands have like complementary information, but sometimes transcription only happens in one direction. And to study this like strand-specific effect, we can also use strand-specific reads. Any questions so far about ChIP-seq? Which paper?
The P part, the P gene. So
Mm-hmm.
that means A could bind to the multiple protein at the same time?
So this is the read. So this is the sequence that binds to one protein. So it's the same protein, but the same protein can bind to different region of the genome. So it's the opposite.
Oh.
Yeah, yeah. Cool. And similarly, this can be used to also measure the histone modifications. As I said, because histones are also DNAs that kind of engaging with, are also proteins that engage with DNA. So we can use similar like antibody to pull them down and observe where they are in the proteins. Yeah. So we could, this example, you have like each fragment's about 30 base pair long. And they map to the unique positions in the 3 billion base reference genomes. So you can see that although our genome is quite, although the vocabulary in our genome is quite poor, only 4 of them, but if you have long enough sequence, you get like 4 to that number of combinations. So in this case, even like a 30 base pair long sequence kind of can get a unique mapping to part of a reference genome, because you basically have like 30. We can consider 30 being kind of already very specific sequence length in this sequencing. And typically this order is in the order of 10 millions. And there's also like you can see like MiSeq, HiSeq, these terminologies, which basically tell you how deep you want to sequence. In this process. Of course, the deeper, the more expensive, or the longer time it takes. And secondly, we can also measure our DNA accessibility or chromatin accessibility by doing things in a similar mindset, but with a different set of proteins, which is called DNase, which is enzyme that can dissolve or kind of cut our DNA if it's a DNA, if the DNA is exposed, is in the loose position and exposed rather than in a compressed form. So we basically add this enzyme into the sample with the right concentration, such that only the part of the gene that it's open and accessible get cut out, and we again can use the same sequencing technique to sequence these reads and map them back to our genome, so we understand like, wow, this of the part of the genome is open in this cell. Very cool. And similarly, there's also other types of technique for measuring the chromatin accessibility, which is called ataxic, which is commonly used, more and more used nowadays. But effectively, they're just using different types of assay and having a kind of more specific adapter DNA that binds and cut the fragments out. And you will get very similar kind of data in terms of their format. But actually, although both of them are detecting chromatin accessibility, they are not always identical, and the correlation between the two is kind of mediocre, like 0.58 with the R square of 0.58. But I think currently what we found is that DNA-seq is a little bit less sensitive than, okay, I'm not edited here, but I think ataxic has a higher we believe that ataxic have like higher accuracy and precisions right now, so we're kind of more frequently using ataxic rather than DNA-seq. And DNA-seq is not a direct evidence of what transcription factor is binding to where, because it only assess like which part of the genome is open. So it would be better if we use it in combination with ChIP-seq to determine like which region is actually doing having a transcription factor binding. And one thing interesting about DNAs is it's hard to find a very specific pattern if you only look at the individual transcription binding site. For example, here is the signal you will get if you only care about a single CTCF. But you cannot get any patterns from this individual CTCF binding, but if you aggregate the binding effect across multiple cells and multiple positions, you will have kind of aggregated feature which kind of tells you what motif is binding to this position in a statistical way. All right, so so the next thing we want to talk about is beyond sequence, right? So we know that things, of course, sequence decides the information of the DNA and part of the protein. However, things are usually more complex, and they have this 3D structure and this spatial pattern that can also indicate some interesting interactions. And we can see that kind of our mammalian cells, it's like this kind of ball where the DNA is located inside the nucleus, which is at the center of the cell. And again, like our cells are super small, and our nucleus is it's even smaller, but it has like a lot of information, which if you extend it, it can be 2 meters long. Yeah, I think this is just interesting. I mean, demonstration of how small our nucleus are comparing to ourselves. And you can see the ratio here is like 6. If you compare like a tennis ball to the nucleus, then sorry. This is the nucleus is the outer side of these pictures, and the centering part is just the DNAs. So if you compare the mass that the DNA takes in the entire nucleus, it will be similar to compare a tennis ball with something that's like 20 kilometer big. So like our DNA is really small, and it's kind of sparsely located in our nucleus, even though the nucleus itself is also very small. Sorry. I apologize for, I think this is a mistake. So what I'm trying to show here is so the thread length is what the actual length of the DNA is, and the diameter is what the size of the nucleus. So yeah, so basically we're compressing the information of 20 kilometers long into this like small like ball of a tennis ball. So that's the rate of compression again. And this thing I mentioned in the beginning of the talk is called DNA compression or DNA compaction. And we know that there's part of, so even though we kind of talk about this histone marks, nucleosomes, and chromatins, but actually we don't understand all of the mechanism of how things get compressed, right? I could talk about how we can locally compact using these histone octamers to form nucleosomes. And we also kind of know that the whole chromosome is compacted locally during some certain phases, but we actually don't know how they get compressed from here to here. And therefore, it kind of motivated the entire study of like, and to understand better about where things are located and how they change in location and how things kind of change from a very loose position into a compact position during different phase of during different cell phase. So yeah, these words don't need to memorize them at all, but just know that kind of in the cell division process, what we call mitosis, there are different stages where kind of our cell prepare itself to become two cells. And then we all know that in this process, like actually in this process, our chromosome has very big changes in where they're located and how compact they are. So at the beginning of this phase called interphase, it's basically a lot of time how the cell is looking like. Our chromosome is basically quite spread out and very sparse. They're kind of loosely opening up in the cell, which makes sense because we want to transcribe and translate, so it cannot be very compact all the time. And then during the stage of cell division, it starts to form this metaphase where you can see this X and Y structure even from a microscopy image, that things get extremely compressed into this X and Y shape chromosome. And then after the division, they kind of spread out again. So there's this interesting field of spatial transcriptomics and genomics, where we can try to measure things by kind of using these fluorescence to track where things are in the cell and different times of the cell, in different timing of the cell division. And out of that, there's a simplified version where besides knowing where they are, we also care, we kind of also just in a high level want to know what is the shape of our chromosome and how the chromosomes, when they collapse and they compressed, how are they folded? It's like protein folding, right? We want to know like if there's certain structure that they always folded into, and how that structure will tell us about how genes are interacting with each other. So there's an area that we call chromosome conformation capturing, or 3C, or anything that with a C in it, where we try to measure the physical interactions between these genomic elements. And the trick here is actually very simple, because if we know that things are contacting, we can try to, again, like freeze things or cross-link things up if they're contacting. And we can cut this small part that are contacting, and we can use, do this like ligation, where things close up into like a single DNA strand, and we can again purify all of these parts that actually form this ligation, and we can again use our sequencing technologies to get this map, and we map it back to the reference genome. Then we know like these two parts of the genome are contacting with each other. And there are actually a wide spectrum of different 3C based methods. It all just depending on whether or not like basically how many pairs and how much throughput you're getting from these assays, and out of which the most kind of very exciting one's called high C. Of course, not this fruit pond, but I think as famous as the fruit pond. But basically, you try to increase the throughput of the assay such that we can do this all-to-all. We can instead of like focusing on only like small region of the chromosome, we can study the entire set universe of chromosome in the nucleus and study their interactions. And what we get from these process is actually something like this pairwise matrix. You can call this adjacency matrix if we're thinking about graph learning. But here we can call it a contact map. But the high-level idea is it tells us the pairwise relationship of how closely things are in the space. And we can see that actually a lot of these interactions are quite local, and basically you don't expect things that are located too further away in the sequence indexing space to fold and contact. It's kind of expected. And there also like kind of this group structure where there's kind of a boundary of each chromosome where things don't cross-react too much beyond that boundary. And CHIA-PAC is just kind of yet another advanced way of studying chromatin interactions. It's instead of doing this kind of ligation, it just try to use kind of again antibodies to pull down things where things are contacting and there is a CTCF binding, and it kind of using this signal to kind of map if two things are contacting and they're also binding to CTCF. And you will be able to get two of them together, and so you will get. And so if you like it together, you either get this self like pairs from the same sequence, or you get pairs of the two contacting sequence, which could be coming from a very remote region. And again, it gives us this kind of contact map type of data, and at the same time, you also get this sequence. Per sequence position readouts, like in ChIP-seq and DNA-seq. And I think CHIA-PAC has a slightly higher resolution than HiSeq, so there are a lot of data generating that's very interesting. All right, so let's talk a little bit more about transcription factors. Since we talk about motifs, I think basically we want to understand motifs better because we know that they're the key patterns that we try to detect when we try to predict transcription factor binding and regulatory activities. So one thing we know that there are like 650,000 known TF motifs, but only but the motifs can bind as we said, like one single transcription factor can bind to multiple positions in the genome. So there are approximately 50,000 binding sites, even for the single, for a typical single transcription factor. So that means we actually have 50,000 copies of sequence that we can use to study like what is contributing to a transcription factor binding, right? So, so a lot of these classical bioinformatics algorithms are just trying to make use of these different copies of transcription factor binding sequence and try to find if there are common patterns from there, such that we can identify this what we call motif. And this is also called the field of motif discovery. So I think a lot of these algorithms are just relying on doing alignment of different sequence because the sequence may not be identical, and sometimes it can be, there's gaps and different small changes in the genome where you need to decide whether these two things are should be aligned from this position or not. So it's kind of a dynamic and iterative process where you kind of have some brief high-level idea of where things align, and then you can get this frequency by aligning things in that way, and then you can try to redo the alignment again by making corrections. So in terms of motifs, there are actually a very particular way of representing motifs, which is what we call position weight matrix, or PWMs. Which you're going to see a lot in the next few lectures. So basically, it's very simple. It's just like once you have these aligned sequences, you compute the frequency of each basis at different position, and then you just normalize it to get a frequency of different bases. So this is what we call the precision probability matrix. And we so you can see that there are some positions where things are very certain, where most of the time it's only one type of the basis, where there could also be places where things are less certain. So for example, in this case, you have 50% of chance being C and G. It particularly could be because of the chemical composition of the two nucleotides are kind of similar, or it's just not exposed, so it does not contribute to specific binding of proteins. So one thing we usually visualize is called this PWM logo, which interesting visualization of this probability where we kind of define the concept of information content, which is the negative of the probability times the log odds between that probability and a background distribution, which basically we believe naturally, randomly, there could be some bias towards certain bases in our genome. So we kind of want to distinguish how much different the current distribution is from the background distribution. And by taking the expectation of these log odds, we kind of compute the information content of this position. And the SIC logo is just a way of, it's basically 2 minus the information content. And it kind of is loosely related to the entropy of that position. So you can see that the total height or number of bits is related to the entropy. The more uncertain you are, the higher you are, the more number of bits. Sorry, the lower you are, the shorter that position is. So the number of bits is smaller. And then you just divide the total hits by the frequency of that amino acid type. So it's kind of a combination of visualizing of how different bases are distributed at each position, also at the same time of visualizing how certain or how low entropy you have in different positions. And then another thing we use is just directly regularize the probability or log odds, where we again take the ratio between the current position weight matrix with the background distribution, which could be a fixed kind of distribution per position, but also it can be position dependent. I don't know. I don't think it's position dependent a lot of time. And then we take the log odds of that, and then we can visualize the logo again here, where things get like kind of negative scale. Here are things that are not being presented a lot, and things got the positive scale here, positive value here are things that are important and differentiating from the background. Okay, so there are also actually a lot of different algorithms that are already introduced to study the motifs. So one key kind of assumption here is that the probability of for each position are independent. So we're not capturing any correlation between different positions by using this position weight matrix, which could be something modeled by, if we use deep learning model, we can definitely model it differently to capture dependency. But I think so far this independent probability matrix has been working pretty well in bioinformatics already, and we can see a lot of clean pattern from just this large sequencing data, and by running any of these more conventional like algorithms that does aligning and probability finding at the same time. But a simple way of using probability PSSM matrix is you just for a given sequence, you can directly compute the PSS score by taking the number of that base at that position and then add them up, right? So you can consider this case, if you represent the sequence into this one hot matrix and you have this PSSM scores, and you will add up the element that is non-zero, and you will get a PSM score here, which tells you how well you matches this motif. And if we do this at different positions, you can get this different numbers, and you can apply a threshold there such that things beyond a certain threshold, you can consider them as positive, like there's a matching motif. And if we think carefully about this, it's actually just exactly like what a convolution filter look like in computer vision. So this is kind of one interesting coincidence that this motif finder is actually can be interpreted as a convolutional filter, and this thresholding step is also can be considered as like activating a neuron in neural networks. So this natural coincidence motivates the use of convolutional neural networks in a lot of these genomic studies, and a lot of these variants are actually so we can basically think of this lower level motifs being the vocabulary of a sequence. And then there's this interaction combinatorial effect between different motifs, where sometimes the sequence can contain more than one binding interface, and sometimes they are binding by multiple transcription factors. So we have this combination of vocabulary, and the vocabularies, when they appear together, they have certain distance, which might vary by what function they perform. So a lot of these high-level combinatorial rules can also be learned with the neural network, considering like this MLP layer on top of the convolutional layers, which try to learn this combinatorial effect. So I think in the summary, like given this kind of similarity, we consider deep convolutional neural networks being kind of a good containing this good inductive bias for studying the regulatory patterns in the genome. So yeah, so from next lecture, we'll be talking about convolutional filters and some advanced modification of that. And the other thing is, you can see that because the only way that we tell that there is a pattern in the sequence is by looking at these motifs, right? So this is also something that a lot of the biologists are familiar with, and it's kind of the signal that they can rely on to interpret what's happening. So this also motivates a lot of the model interpretability tools, which try to visualize these motifs as a result. Cool. So yeah, I don't think we have time for the gene expression, but we will definitely cover it on the second lecture of regulatory genomics. Any questions for today? Sir, can you repeat that with louder voice?
So we have these models where we're looking for motifs.
Mm-hmm. Yeah.
If we could discover, let's say, like a complete list of all the motifs
Mm-hmm.
that are used in the genome,
Yeah.
what would that enable in terms of applications in terms of pharmaceuticals?
Yeah, that is super interesting question. I believe it has a lot of potentials. The first is definitely understanding like the mechanism behind how things are regulated. The other thing you can easily do is, think about therapeutics, right? We can kind of actively, because we have these cool technologies of editing our genomes and doing these interventions, we can think about like gene therapies where we try to target these specific patterns and we try to correct things up if we know what's going wrong in there. And we can also come up with different combinations of enhancer promoters, and so we can use them to regulate the expression of very important